Bismillahirrahmanirrahim. In our previous lectures, we discussed the cellular adaptations, the responses of the cell to injurious stimuli. We discussed reversible and irreversible cell injury. We discussed various causes that cause cell injury, and we also discussed the necrosis and various patterns of the tissue necrosis. Today we are going to discuss briefly the mechanism of cell injury. Before going into the various mechanisms that are involved in cell injury, here it is important to say that each individual is unique. Each person differs from the other in their response to injurious stimuli, and in the same way, different individuals have different disease predisposition, and also they differ in response to the environmental agents, and all these differences arise from the genetic makeup of an individual. We will study in genetics that genetic polymorphism, in which an amino acid in the genetic sequence is substituted by another amino acid, and this type of polymorphism is seen in many people. And individuals with various genetic polymorphism also differ in their response to environmental agents, in their disease predisposition, in the response to injurious stimuli, and also the response to drugs, to various medicines. And a research is going on to establish personalized medicine known as the pharmacogenomics, in which an individual with a particular genetic makeup will respond to a drug quite different from another individual. Moreover, the diversities that arise as a result of genetic polymorphism in various metabolic pathways, and along with the genetic polymorphisms leading to various diversities in the metabolic pathways, constitute the response of an individual to a particular stimulus or to a particular drug in combination with the environmental factors. So each and every disease and its effects on a particular individual are determined by the genetic predisposition and the environmental factors. For example, a person who, when exposed to particular environmental factors, for example, sedentary life, high fat diets, and other determinants, he will develop hypertension. But the same environmental factors may not produce hypertension in other individuals. The same can be true about diabetes mellitus and so many other diseases. So the development of a disease in a particular individual depends on the genetic makeup of the person plus the environmental factors. Now, the determinants of the cell injury depends on the injury as well as on the cell type affected. The type of injury the duration for which the injury lasts, and the severity of the injury will determine the outcome in a particular individual. For example, ischemia and hypoxia that stay for shorter durations, and if they are of mild to moderate severity, can result in reversible cell injury. The same is true about the small doses of a toxin. But the severe injury lasting for longer periods may result in irreversible injury. So, the determinants of the cell injury, and particularly when we talk about the injury, depends on the type of the injury, the duration for which the injury lasts, and the severity of the injury. Now, on the other end of the parameter, the type of the cell, the status, adaptability, the ability to adapt itself, and the genetic makeup of the cell will also determine the outcome of an injury. For example, the skeletal muscles cells, they can tolerate ischemia for about hours, while the cardiac muscle cells die within 20 to 30 minutes, and the cells of the nervous system die more quickly. So the type of the cell, if it is a skeletal muscle cell, it will sustain injury for longer periods than if it is a cardiac myocyte or a nervous cell. The status of the cell, for example, when the oxidative phosphorylation is disturbed, that usually results from ischemia and hypoxia, the cell will use another way for generating ATP, and that is the anaerobic glycolysis. And that will lead to the use of glycogen to be converted into glucose. So the status of a hepatocyte liver cell having enough glycogen will sustain anaerobic glycolysis longer than the cell that has no glycogen. The adaptability of the cells, for example, we can say that certain cells can undergo hypertrophy but no hyperplasia. For example, the stable cells of the glandular tissue will undergo hypertrophy and hyperplasia, and the skeletal muscle cells, cardiac muscle cells, they will undergo hypertrophy, but there is no hyperplasia. And moreover, the cells of the central nervous system, which are considered to be the permanent cells, they once injured, they have no way to return to normalcy. And the genetic makeup of a person and of a cell also determines the outcome that result from a particular injury to a particular cell. Now, coming to the mechanisms of cell injury, the mechanism of the cell injury and tissue injury is mostly very complicated, and the combination of various defects give rise to the outcome of the injury. But the well-known factors are the ATP depletion. ATP is continually produced in the mitochondria, which is the energy house of the cell, and this ATP is needed by many metabolic processes inside a cell. The ATP depletion can also occur in case of mitochondrial damage, and moreover, the influx of calcium into the cell that usually results from defect in the membrane permeability will result in activation of various proteolytic enzymes. Normally, the reactive oxygen species are produced during oxidative phosphorylation, but there are compensatory mechanisms as antioxidants and they neutralize these reactive oxygen species. But if the reactive oxygen species are produced much more that can be neutralized in pathological processes, they will accumulate and will cause serious damages to the various components of the cells, particularly the cell membrane phospholipids, the proteins, DNA, RNA, and various genetic apparatus can be damaged. The defects in membranes, particularly the cell plasma membrane, the membranes of the organelles like lysosomes, the Golgi apparatus, the endoplasmic reticulum, play important role in cell injury. And finally, the damage to the nuclear apparatus like DNA and various structural and functional proteins play important role in the cell injury. So routinely the cell pass through a reversible cell injury, and if the injuring agent is removed, there is a chance for the cell to become normal again. But if the injury is so severe and or if there is an inherited defect in a cell, it will undergo irreversible cell injury in the form of either necrosis and apoptosis, which is a type of suicidal cell death that we will discuss in detail later on. Normally, in a cell under physiological conditions, the ADP is phosphorylated in the availability in the presence of oxygen by the oxidation of proteins, carbohydrates, and fats. And this ATP will be used for various cellular processes. In case of mitochondrial damage or ischemia or hypoxia, what will happen that the alternate way for the generation of ATP is the anaerobic glycolysis that will occur in the absence of oxygen. This anaerobic glycolysis gives a limited supply of ATP to the cell for their metabolic functions, but it is a short-lived process. And the effects of anaerobic glycolysis that we are going to discuss will cause the cell death. The ATP that is generated by the oxidation of proteins, carbohydrates, and fats by phosphorylating the ADP, this ATP will be used by various cellular processes. For example, the active ion transport along the membranes, the muscle contraction, and the synthesis of certain molecules, and the same ATP will be used for cell division and growth. Now, a few things that are comparatively well known and that take part in the cell injury is the mitochondrial damage. Whenever the mitochondria is damaged, we know that mitochondria is the site of ATP production. And when the mitochondria is damaged, there will be decreased ATP production or ATP depletion. And since we know that this ATP is needed by the energy dependent cellular functions, so when there is no ATP, these energy dependent cellular functions are deranged, and ultimately it will culminate in necrosis, and sometimes it will lead to the suicidal cell death or apoptosis, in which the various mitochondrial proteins are released, the known as the pro-apoptic and anti-apoptic proteins. And when the pro-apoptic proteins are greater and stronger in amount than anti-apoptic proteins, it will lead to the apoptic cell death. The other component that takes important part in cellular damage is the increased permeability of the cellular membranes, particularly the plasma membrane, lysosomal membranes, and mitochondrial membranes and membranes of the endoplasmic reticulum. So what happens that the ionic transport across these membranes is mostly ATP dependent, energy dependent, and when there is no ATP due to mitochondrial damage caused by hypoxia, ischemia, or toxins, what will happen that the ionic transport across these lysosomal membranes and the transport of water will be deranged, and it will lead to its detrimental consequences. Normally, the calcium level inside the cell and outside the cell is maintained by the active calcium transport system. But if the membranes are defected, and in cases when there is damage to the mitochondria and the rough endoplasmic reticulum, what will happen that the cytosolic calcium level will increase, and this calcium will activate various proteins, proteolytic enzymes like proteases, and they may cause the degradation of the cellular components. And sometimes this influx of calcium and increased level of calcium inside the cell may lead to the apoptic cell death. The reactive oxygen species that are generated in the electron transport system during the production of ATP by the oxidative phosphorylation, normally these reactive oxygen species are neutralized by the antioxidant compensatory mechanisms. But if the antioxidant mechanisms fail to neutralize these reactive oxygen species, or if the production of reactive oxygen species is very more in various pathological conditions, then what will happen that these reactive oxygen species, they will accumulate inside the cell, and they will damage the cellular proteins, structural and functional proteins. They will also damage the lipids, particularly the phospholipid bilayers of the membranes, and also the nucleic acids, DNA and RNA. The damaged DNA and misfolded proteins, they are normally removed by the DNA repairing mechanisms and the presence of chaperones. But if the neutralizing mechanism is weaker, or if the production of the damaged DNA and misfolded proteins is more than they can be neutralized by the chaperones and other mechanisms, what will happen that the accumulation of the damaged DNA and misfolded proteins, they will activate the apoptic cell death. Now, here we discuss the damage to mitochondria, which may be caused most commonly by ischemia and ischemia leading to hypoxia. Here I want to tell my students that the difference between ischemia and hypoxia is that the ischemia is the decreased blood supply to a particular tissue, while hypoxia means decreased oxygenation of the tissue. So ischemia can cause hypoxia. Hypoxia, the most common cause of hypoxia is ischemia, but hypoxia can also occur when there is no ischemia, but if there is defective oxygenation of the blood, for example in various lung diseases like pneumonia and other, in which the oxygenation of the blood is defective, and though there is no ischemia, but still the tissue suffer from deficiency of the oxygen. So when there is deficiency of oxygen, the oxidative phosphorylation in the mitochondria, which is the major source of ATP generation, it will become defective. And when the absence of oxygen cause defective or decreased effective oxidative phosphorylation, there will be decreased generation of ATP. Now we know that this ATP is required by the sodium potassium pump. It is required by the calcium pump. And what will happen that when there is no ATP, the sodium, it will rush into the inside of the cell followed by water. Osmotic gain of water inside the cell and cell organelles. Normally during the sodium potassium ATPase pump, what happens that 3 sodiums are pushed out into the extracellular space, while 2 potassiums are pumped inside into the cell. So what will happen that when the unavailability of the ATP will cause damage to the sodium potassium ATPase pump. What will happen that the sodium will rush into the inside of the water and followed by the osmotic gain of water, and the calcium pumps are damaged, and what will happen that there will be increased influx of calcium and, as I said, sodium followed by water, and there will be increased efflux of potassium when the sodium potassium ATPase pump is deranged. So what will happen that the net gain of water will lead to endoplasmic reticular swelling, cellular swelling, and as a result of the cellular swelling there will be loss of the microvilli and formation of blebs in the cell plasma membrane. Moreover, when there is no when the ATP generation is decreased through oxidative phosphorylation, what happens that the cell will try to get energy from the anaerobic glycolysis, in which the glucose derived from the glycogen is uh, glycolized, and in the absence of oxygen it will for a short period of time it will give limited production of ATP, but as this we know that this process is a short-lived process and it will lead to depletion of the glycogen stores. As a result of anaerobic glycolysis, there will be accumulation of lactic acid, and it will decrease the pH of the blood. The blood will be the tissue and the intracellular compartment will become more acidic, and it will lead to the various inactivation of the enzymes that are needed for a normal cell function. Moreover, decreased in ATP generation will lead to the detachment of ribosomes from the rough endoplasmic reticulum. Normally, the ribosomes, they are attached to the rough endoplasmic reticulum, and these are the stores for protein synthesis. Various structural and functional proteins are found in the ribosomes. But when the ATP is depleted or if it is decreased, what will happen that these ribosomes, they will get detached from the swollen rough endoplasmic reticulum, and ultimately it will lead to a decrease in protein synthesis. Now, here in this slide, we discuss various targets of the cell injury. First of all, we discuss the mitochondrial damage. And as we said that the mitochondria, which are the main sources of ATP generation through oxidative phosphorylation, what will happen that decreased production of ATP that will occur in hypoxia and certain toxins causing damage to the mitochondria will lead to the decreased generation of ATP, and with its multiple downstream effects that we have discussed and we will discuss a little in detail in future. The mitochondrial damage will result in the increased reactive oxygen species production. As I have said that normally the reactive oxygen species that are produced during the electron transport system and during oxidative phosphorylation, they are neutralized by the antioxidant mechanisms. But excess production of reactive oxygen species in pathological processes, it will cause damage to the lipids, particularly the phospholipid bilayers of the cell membranes, to the structural and functional proteins, and damage to the DNA. Now, the second target of the cell injury is the membrane damage. Now, this membrane is the cell plasma membrane, the membranes of the organelles like lysosomes, endoplasmic reticulum, and nuclear membrane. So, what will happen that when the cell plasma membrane is damaged, it will lead to the loss of cellular components. When the lysosomal membrane is damaged, what happens that various proteolytic enzymes and hydrolytic enzymes, they are released, and they will cause enzymatic digestion of the cellular components. Now, the third mechanism of the cell injury is the influx of calcium. When the calcium transport system is damaged, which also requires ATP, what will happen that it will lead to increased in mitochondrial permeability pores, and the increased intracellular level of the calcium, it will lead to activation of the multiple cellular enzymes, proteolytic enzymes, hydrolytic enzymes, and various enzymes like endonucleases, and they have their detrimental effects. The protein folding is a process in which the various amino acids are collected and structured together in a 3D uh, uh, fashion that will lead to the formation of a particular specific active proteins. But if the amino acids are not properly folded, it will, this we say is the protein misfolding, and these misfolding proteins, they will accumulate. Normally the proteosomes and the chaperones, they will compete to they will try to prevent the misfolding of proteins and to the proteolysis of the damaged proteins. But if the production of misfolded protein is more, or if there is an inherited damage to the DNA, what will happen that the from the mitochondria the pro-apoptic proteins are released, and the anti-apoptic mechanisms, they comparatively becomes weak against the pro-apoptic proteins, and these pro-apoptic proteins, they will activate the apoptic cell death. Here again we revise the mitochondrial damage, which is mostly caused by the decreased oxygen supply, ischemia, hypoxia, toxins, and sometimes by radiations. And the mitochondrial damage will result in decreased ATP generation and increased production of the reactive oxygen species, and both of these will lead to multiple cellular abnormalities, and that will ultimately lead to the irreversible necrotic cell death. Apoptosis, we will discuss apoptic cell death and the mechanism of apoptosis in detail in our next coming lectures. But here simply we can say that the survival signals, when the cells are derived of the survival signals, or if there is damage to the DNA and accumulation of misfolded proteins, it will lead to the activation of pro-apoptic proteins and depression of the anti-apoptic mechanisms. And what will happen that the pro-apoptic proteins, they are released, and they will cause the apoptic cell death, the mechanism that we will discuss in more detail in our lecture on apoptosis. Now, the influx of calcium has a very important role in cell injury. And here I want to say that there are three sources of calcium from which the calcium is derived are the extracellular calcium, particularly when the calcium transport system is damaged, and also the calcium is normally sequestered within the endoplasmic reticulum and within the mitochondria. But in case of damage, these three sources of oxygen, the three sources of calcium will lead to increased cytosolic level of the calcium, and this increased cytosolic calcium will cause activation of the cellular enzymes. For example, the phospholipases are activated, that will degrade the membrane phospholipids, particularly the phospholipid bilayer of the membranes. The proteases, they are activated, they will cause disruption of the membrane and cytoskeletal proteins. The endonucleases are activated, which will degrade the nuclear damage, and the proteases and phospholipase activation will lead to membrane damage, while the activation of the endonucleases will lead to the nuclear damage. Moreover, the enzyme ATPase, they degrade ATP. If it is activated, it will degrade the ATP, and already the ATP is less due to the mitochondrial damage, and this super added ATPase activity will cause further depletion of the ATP, and the mitochondrial damage will result in the formation of the mitochondrial permeability transition pores. Now, the membrane damage has very important role in cell injury. And what happens that the intracellular influx of calcium will lead to the activation of phospholipases that will cause phospholipid degradation and membrane various membranes damaging effect. The proteases are activated, and these proteases, they will degrade the cytoskeletal proteins, and the reactive oxygen species, they have their effect on lipid peroxidation, also on the DNA proteins and other structural and functional proteins. And the loss of phospholipid as a result of the decreased ATP and as a result of the increased influx of calcium will lead to loss of the phospholipid, and the membrane phospholipid bilayer is damaged. Moreover, the phospholipid degradation, which is caused by the phospholipases and which are activated by the increased cytosolic calcium level, will lead to the generation of lipid breakdown products, and they also will injure the membrane and the membranes of the plasma membranes of the cell organelles, nucleus, rough endoplasmic reticulum, and Golgi apparatus, and other organelle membranes. The proteases which are activated, they will degrade the cytoskeletal proteins, and they are activated by the increased influx of calcium, and ultimately these three processes, loss of phospholipid, the detrimental effects of lipid breakdown products, and the cytoskeletal damage caused by the proteases, they will lead to the membrane damage. So coming to a summary of that, what we discussed in the mechanism of cell injury, number one is the ATP depletion. Automatically, when there is no ATP, multiple processes that occur inside the cell, for example, ionic transport and sodium potassium ATPase pump, calcium transport. So all these energy dependent functions are becomes defective, and they will cause the irreversible injury to the cell that will culminate in necrotic cell death. The damage to the mitochondria will lead to ATP depletion, and followed by the ATP dependent cellular functional derangement, and may lead to necrotic or apoptic cell death. As we said, the influx of calcium has very important role, and it will activate various enzymes, and that will lead to the necrotic or apoptic cell death. The reactive oxygen species, we will discuss the reactive oxygen species in my next lecture, and how they are generated and how do they are neutralized, and when and where they have their detrimental effects on proteins, lipids, and nucleic acids. The defects in the permeability of various membranes, for example, plasma membrane, lysosomal membranes, mitochondrial membranes, when they becomes defective, and if the ionic transport mechanisms are deranged, ultimately they will lead to the necrotic cell death. And finally, the accumulation of damaged DNA. Normally, when the DNA is damaged, there is a mechanism in the cell, that is the DNA repairing process. And if the DNA can be repaired, it is repaired, but if the damage is severe, the cells are directed to undergo apoptic cell death. And in the similar way, the accumulation of misfolded proteins along with the accumulation of damaged DNA will trigger the apoptic cell death. Thank you.